Hello, my name is Raymond Caldera, and today I am presenting to you my final project, Genetic Intervention, a historical advancement in pest control. Published only last month by the University of Virginia Tech, this study conducted contributes to our options for genetic intervention in mosquito populations. By identifying new targets for genetic editing, scientists have a new option for mosquito control, which has proven in this study to result in the sterilization of 10% of the total offspring generations in this trial. This was accomplished by targeting pathway genes that determine sex, effectively disrupting anatomical development and ultimately disabling reproductivity in a portion of the offspring. With the success of this study, scientists are now closer to effectively combating the spread of disease such as Zika and dengue fever. It is important to recognize that the accomplishments of the study conducted by Virginia Tech were not merely invoked out of sudden opportunism, but was instead one monumental step in a greater history of efforts to overcome the struggles of pests and insects. Throughout history, mankind has continuously fallen victim to issues stemming from insects. The most common struggle, characterized by insects, consists of the spread of disease and the damage and predation of our crops and livestock. Many methods have been proposed and employed throughout recent scientific efforts. This has often consisted of external chemical agents such as pesticides, but like the methods conducted by Virginia Tech, population control by means of reproduction control has also persisted as a means of attack. One of our earliest methods that aimed to control pest populations by means of fertility came in the 1930s, where scientists began to experiment with the use of radiation for such purposes. By effectively utilizing ionized radiation in male populations of insects, we became capable of creating a generation of sterile males that could potentially be released in a wild environment to crash successive generations of offspring. This method would find its first large-scale success in 1954, where the method was employed on a small island just off the coast of Venezuela named Curacao. In only 7 weeks' time, the entire population of screwworms, a species of flies notorious for preying on local goat herds, would be entirely wiped off the island. By the 1960s, we began to see some of the first great advancements in genetic research for alternative ways to hinder insect reproduction. This new effort in genetic intervention would be largely characterized by advancements in the field of what has become termed as gene drives. Counter to typical Mendelian inheritance, in which beneficial alleles may spread through a population due to their beneficial contribution to the survival and subsequent reproduction of the organism, a gene drive would instead relay alleles to offspring in an interaction in which a portion of the gene is severed by a microbe and replaced by a copy of the microbe's own DNA. What this can allow for is the passing of non-beneficial genes that would oftentimes be filtered out of inheritance due to potential negative effects to the survival or reproduction of that organism. Although efforts to utilize this microbial mechanism to our advantage would struggle to become widely applicable for those purposes of insect population control, due to the challenges in being able to intentionally control the targeting of desirable genes for replacement. This would largely remain the case until the 1990s, in which new advancements in our research of endonuclease-based gene drives would finally enable us to control the targeting at which genes would be severed and replaced. Gene editing today may be categorized in four primary groups: MN, TALENs, ZFN, and CRISPR. The latest and greatest of these groups is CRISPR, which was invented in 2012 by the scientists listed below. It's in this context of the advancements of gene editing made within the last century, and especially CRISPR, that has warranted great optimism and efforts of teams such as the group at Virginia Tech. Their work to identify genes responsible for sex determination in mosquitoes with the goal of controlling reproduction and ultimately reducing disease contractions is directly enabled by our historic efforts to advance gene editing. Lastly, as our world continues to change and challenges evolve due to factors such as climate change, the research of teams such as that conducted by Virginia Tech will only become more necessary. Mosquito and insect populations are expected to rise with warmer climates, and gene editing may offer a cleaner solution than traditional insecticides, which may be harmful to the environment and non intended targets.